Hey, hi, good afternoon, students. I'm Miss Yo. Okay, and today I'll be going through your Combined Science Bio 2017 practical exam paper. Okay, this is a paper that y'all did quite a few weeks ago already. So just to freshen up your memory, okay, in case y'all forget, this is the experiment that y'all did. Okay, y'all have oh wait, I forgot to share screen. Hold on. Let me share screen. Okay, so yes, okay, I can see. Okay, yes, now we have the screen sharing on. Okay, so this is the experiment that y'all did a few weeks ago, and this is the question paper, and it's basically an experiment where you have to find out, okay, how the potato changes its length after you place it in two solutions A and B. Okay, so the first thing that you'll see when you go into the lab is this. Hold on. Okay, so you'll see this experimental setup. So you have two solutions, A and B, with their respective test tubes. Okay, it's a little bit small, but I hope you can see and remember what you did. Okay, then you have your potato together with the measuring towel, subscaffold, and then the ruler. Okay, and what they want you to do for this experiment is, firstly, they want you to fill the boiling tube A and B. Okay, half of the boiling tube A and B with the liquid A and B. See, so the keyword here is actually approximately. Okay, so when they say approximately, it really means about half. So you don't have to really spend too much time to try and measure that test tube and then find the halfway mark and then fill a liquid, okay? So you really just need to do about half of volume tube A and half of volume tube B with their respective solutions. But one thing that you must note, so one thing that you must note is that after you fill boiling tubes A and B with their solutions, the volume of the solution must be the same in both boiling tubes. Okay, so it can be a little bit more than half, but just need to ensure that at the end, the amount of solution in A and B is the same. Okay, so that's the more important point lah, rather than half. Okay, then after that, they want you to cut your sweet potato into approximately 5 mm thickness and 40 to 60 mm long. Okay, and then before that, just a little tip, okay, for you all to more easily measure the width, the 5 mm width, because sometimes with a potato that is round, circular, it's not flat. Okay, so it's a little bit hard to measure the width if you want to try and use your ruler to directly measure the potato. So one thing that y'all can actually do is this. So again, it's a bit small. Let me try it. Okay, yes. So if you can see, I actually placed, Mr. Cho actually placed the towel on the just on the table, okay, and then he placed the ruler against the towel. So the 0 cm or the 0 mm of the ruler is against the starting of the towel, okay, against the beginning of the towel, and then yeah, he just rests the ruler against your towel. And from this, you are able to more easily measure your potato because you are able to just place your potato on the towel as how you usually would, okay, and then make sure to just place the potato towards the edge of the towel, towards the 0 centimeters on your ruler, and then just straight away measure from there. So it's a lot easier because the towel is flat, right? It's straight, it's flat, so it's a lot easier to measure out 5 mm rather than using the potato directly. So this is just a little tip that helps you to more easily measure out width of a circular object like a potato. Okay, then after that, they want you to cut it into 40 to 60 mm long strips and then no skin left on it because the skin creates this additional layer of barrier that we don't want. Okay, and then eventually you get potato strips like this, basically this I mentioned, and then you want to dry the potato strips with filter paper. And before putting your potato strips into your solutions, they want you to measure the longest side of the potato stick to the nearest mm, okay, for both potato strips. So the unit here is of course important because if you put it in cm, then it will be wrong. Okay, it will just be wrong automatically. So yes, before you put in the solution, measure the longest side of your potato strips. Then you put it into your solution, ensuring that the liquid actually covers the entire potato. So again, if you top up one test tube, remember to top up the other so that the end volume is the same for both test tubes. Okay, then you wait for 25 minutes. And then after 25 minutes, you can remove the potato blot them dry, okay, and then again measure their longest length to the nearest mm. Okay, then you want to basically, sorry for that description, okay, and sorry, let me just delete them first. Okay. Okay, so basically, you want to basically calculate or record down your results in this table. So the start, the potato length should be about the same for everyone. It should be about 50 mm, okay, or 40 to 60 mm. So it can be anywhere in between. For now, we just use 50 mm, okay, and because you're a Sec 4, you should already know that in your table, only the headings should have your units. Okay, so whatever that's in your table should not have any units. Okay, so both potato sizes should be around the same length, okay, at the start. And 25 minutes later, okay, you will realize that the potato actually changed in length. Okay, and the difference in length for potato A should be an increase. Okay, it should be an increase. Okay, let me just put it down now. It should be an increase, whereas for potato B, it should be a decrease. So for mine, it increased by 2 mm for potato A and decreased by 2 mm for potato B. So the final length after 25 minutes for potato A will be about 52, and then for potato B will be about 48. Okay, so if you have this increasing length, this trend, okay, for your potato sticks A and B, then that should be good lah. Okay, and then for the next part, they just want you to describe one other difference apart from this change in length. So if you are able to feel the potato, right, you realize that your potato A actually feels turgid, okay, whereas for B it feels flaccid. Okay, so that is the answer. So you can just write, let me change to blue color. Okay, you can just write potato stick A becomes turgid, which should be a term that you are very familiar with. Okay, if your plants, you cannot say, these are plants, you must always remember the word turgid and flaccid. Okay, while potato stick B becomes flaccid. Okay, so that's simple, right? It's one mark. It's just one becomes turgid, one becomes flaccid. Okay, and then the next part is the part that they want you to basically explain the differences that you see. Okay, so the differences can be both the turgid and flaccid part as well as the change in length that you observe in the initial experiment. Okay, so if you realize that your potato A, it increases in length, becomes turgid, you should already know, think about the concept of osmosis. Okay, you should know that osmosis occurs and there's a net movement of water molecules or just water into the potato stick A, and that's why it creates this turgid, it feels turgid and there's an increase in length. Okay, so that's actually the answer for this. And then the opposite happens for potato stick B. That's why it becomes flaccid and there is a decrease in the length. Okay, so let me just type it down. For potato stick A, this is what will happen. Okay. Okay, so for potato stick A, we can say that the solution A has a higher water potential. Again, I don't ever write water concentration because it's not the correct term. It's water potential. There's no such thing as water concentration. Okay, so solution A has a higher water potential than the cells, our bracket cell set, okay, of potato A. And because of this, there will be or there is a net, shorten this so that there is a net movement of water molecules from solution A into potato A through a partially permeable membrane via osmosis. So the part here, okay, through a partially permeable membrane via osmosis is very important. Okay, you must always remember to state the process, okay, the process that occurred, whether it's osmosis in this case or like diffusion or active transport. It's very important to state the process because I mean, that will give you the mark lah. We want to know that, this is the concept of osmosis. Okay. And that is why there is an increase in length, okay, of potato A, and it becomes turgid. Okay, so that's done. That's the explanation done for your potato stick A. For potato stick B, it's just really the opposite. Okay, that's why you observe opposite observations, right? So for potato stick B, why it becomes flaccid and why it decreases in length is because solution B has a lower water potential than the cells or the cell step of potato B. Okay, because of this, there is a net movement of water molecules from potato B into solution B through a partially permeable membrane via osmosis. Again, these two answers are very similar. You just have to change the crucial words. Okay, and as a result, there is a decrease in the length of potato B, and it becomes flaccid. Okay, so again, keyword higher or lower water potential, and the movement. Okay, the next keyword is obviously movement of water molecules, the direction or the net movement of water molecules. Where does it move from? Okay, and then you must also clearly explicitly state the process, osmosis and partially permeable membrane. Okay, let me just, if this is easier for you, can you just take a screenshot of this, because I know the lines at the back makes it a little bit hard to see. Okay, so let me just highlight it so that you can take a screenshot and then copy it down slowly. Okay, as I move on to the next part. Okay, the next part, they want to ask you to suggest a way for your experiment to become more accurate. Okay, this is quite a standard form of questioning. Okay, it usually comes on almost every practical, or if not all practicals so far. Okay, and the answer is also quite standard. So if you can, go ahead and memorize this answer, or you should already know actually. Okay, so the way that you can make your experiments more accurate is to repeat. Okay, let me change the color and move it down. Is to repeat the experiment 3 more times. And once you repeat this experiment 3 times, usually we'll repeat the experiment 3 times. 3 is like the magic number, okay? So we'll repeat the experiment at least 3 more times, and then we would calculate the average change in length. Again, minimize this, okay? And then calculate the average change in length of the potato. This will give us a more accurate and reliable result. Okay, so repeating the experiment 3 more times is like the thing that we always do to ensure that our investigation can be more accurate. Because when we repeat it more, we can always calculate the average. Okay, in this case, the average change in length of the potatoes. Okay, crazy, right? Okay, and then the next part, they say that they provide you with identical sticks of another vegetable, and then ask you how you can determine the concentration of the cell sets within the cells of this vegetable. So basically, this question is asking you how is a planning question. So you have to plan for an experiment to basically determine the concentration of the cell set, okay, of this vegetable that they give you. Okay, so how you can do this is usually, we always, in every experiment, not usually, sorry, in every experiment, we always have an independent variable. That means the one that we change, and a dependent variable, the one that is being affected by the change in your independent variable. Okay, so for this kind of question, the independent variable would be the concentration of solution, okay, that we use. So in this case, we can use sugar solutions, okay, and then change the concentration of our sugar solution and then see how these different concentrations can affect our dependent variable, which is the length of our vegetable. Okay, the length of the sticks of our vegetable in the end. Okay, so I will just write the answer down. Okay, so we have, like I said, we have our independent variable. So we can write that down first. Okay, prepare 5 solutions. Usually we use 5 as the minimum number of independent variables to have. So like because 5 is usually it gives us a more holistic picture of our experiment. Okay, so we prepare 5 solutions with different, like I mentioned just now, right, different sugar concentration. In this case, I think we have to specify, okay, the sugar that we want to use. So usually we'll use sucrose. Okay, so prepare 5 solutions with sucrose concentration of 0, 1, 2, 3, and 4 mole per dm cube. Okay, and having a zero concentration is very important. Okay, every experiment should always have a zero concentration of something. So, I mean, in this case, it will be a zero concentration of sucrose, because that means if you have a zero concentration of sucrose, means that it's just pure water. Okay, so if you have pure water and you put your potato cells or potato sticks in them, you can already predict some results. Okay, so having a zero concentration where you already can predict the results of is always good to have in your experiment. Okay, so zero concentration is important. Okay, then the next step would be to place 3 sticks of these vegetables into each solution for 25 minutes. Okay, so the reason why I use 3 sticks for each solution is because over here, right, in the previous question, I just said how to make my experiment more accurate is to actually repeat the experiment 3 times. So just having 3 sticks of vegetable is as effective as repeating the experiment 3 times, because you are using 3 different sticks of vegetables. Okay, and then for 25 minutes, because I just stick the information from the experiment that I already did. So one thing good about planning questions, right, is that they'll usually give you very similar scenarios to the experiment that you did. So you can kind of pick information from the actual experiment and then place it into your answer. Okay. And then after my 25 minutes, I will blot dry and then measure the average change in length of the vegetables. Okay, so I would again measure the average change in length. Since I did 3 times, so I have to measure the average change in length. Then this part is maybe some of the part that y'all did not get. So I will plot a graph. Okay, my graph, let me just expand a little bit on the graph. Okay, so I'll plot a graph. Okay, the independent variable, okay, or the independent variable, which is your y-axis, okay, will be my, independent variable, which is your x-axis, okay, will be my sucrose concentration, okay, and then my dependent variable, the one that changes or is affected by, okay, my dependent variable will be my x-axis, right? Okay, let me just change. Okay. Dependent variable will be my x-axis. My God, sorry, my y-axis. Okay, and then this would be my average change in length. Okay, and then the next step that I want to do, okay, is I want to find the x-intercept. Okay, let me illustrate to you, okay, what I mean by finding the x-intercept and why it's important. Okay, so I just draw here. Okay, so I did mention, right, that I want my graph x-axis, which you should know is the horizontal one, okay, to be my independent variable. So to be my sucrose concentration, okay, per mole per dm cube. Okay, so this, you don't have to draw the graph, okay, as your answer, but just to illustrate to you why finding the x-intercept is so important. Okay, and then my y-axis, okay, will be my average change in length. Usually we will just follow the experiment that was already given to us. So we'll calculate it in mm. Okay, so I know that just now I say my sucrose solution, I have 0, 1, 2, 3, and 4, right? Mole per dm cube. So let's say my average change in length is 0, followed by, plus 1, plus 2, plus 3, plus 4 mm. Okay, let's just say this is hypothetical. So if I have an x-intercept, okay, it means that it passes through my x, right? It can be here, or it can be here, it can be here, wherever, depending on your plot and depending on your results. Okay, but what's important is that, okay, let me see. What's important is that when I have my x-intercept, right, this is your x-axis, in case y'all didn't know, y'all should know, okay, and this is your y-axis. So what's important for me when I find my x-intercept is that when I find my x-intercept, I realize that my x-intercept is when my average change in length is actually 0. Okay, which means there's no change. The average change in length is 0, so there's no change in length. So when there's no change in length, I can already know that based on my concept of osmosis, that there will be no net movement of water molecules into or out of the vegetable. Okay, osmosis still occurs. So let's not misunderstand. Osmosis still occurs. So water molecules still move in and out, but there is no net movement of water molecules. So that means the rate of water moving into the vegetable and the rate of water moving out of the vegetable is equal. Okay, so that's why there's no change in your length at all. Okay, so finding the x-intercept is important. Okay, and then the final part of our answer is to explain why finding the x-intercept is so important. Okay, why it's important is because this will be the concentration at concentration at which the solution has equal water potential as the cell set. Okay, so again, y'all can screenshot this once I highlight so that it's not so messy with the background lines. Okay. And then next, okay, the next part is just biological drawings, okay? There are 3 points for this, even though it's only 2 marks. Even though it's only 2 marks, but there are 3 points to this drawing on how you can score the full 2 marks. Okay, first, you must make your drawing as close as possible to the picture. So like even the number of P's inside, right, must be the same and include this like little half a P over here. Okay, so it must be as close as possible to the picture that they give you. That's the first one. The second one, the size of it. So the size of your drawing must occupy at least 75 to 80% of this box that they give. It's even better if you get 90%, okay, of the box filled. And the last one is your lines. So your lines must be drawn cleanly. So I don't see any sketches, okay, to try and draw the nicest diagram ever, okay? So your lines must be clean and precise. And that's basically how you can get full marks. Okay, and then now we move on. Okay, so they want you to measure the distance between X and Z on the photograph. So it should be about 6.9. It should be about 6.9 cm if you measure correctly. Okay, and then the distance between the same points on your diagram, so on your drawing. Again, if you draw it big enough, the diagram or your drawing should be, the distance between X and Z on your diagram should be more than 9.0 if you draw it big enough lah. Okay, and then the last part, they just want you to basically find the magnification. Okay, so the magnification is basically the magnification of your drawing. So it's the size of your drawing or the length of your drawing divided by the length of the picture. Okay, and the important thing to note here is that you must leave your answer to 1 dp, and the units for magnification is always x. Okay, x for times. Okay, so if you leave out the dp or you leave out the x, you'll get 0 marks. Okay, and the last, and I think, yes, the last question, they're just asking you that a student wants to draw a more accurate biological diagram of the structure of a root hair. Okay, and root hairs, as you all know, is microscopic. Okay, it's super small, so you cannot see it with your naked eye. So in order for him to be able to see it and then draw it, the additional apparatus that he needs will be a microscope. Okay, so easy, it's just a microscope. Okay, and why does he need it? I actually already told you the answer. Okay, it's because the root hair, okay, let me change the blue. The root hair is not visible to the naked eye. Okay, so that's why he needs a microscope. That's why he needs a microscope to view, okay, to view the root hair to draw an accurate diagram. Okay, so that's basically it. He just needs a microscope because it's not visible to the naked eye. Okay. So this experiment is there's a lot of common questions that will be asked, especially for, I mean, actually all of these questions are very commonly asked. You'll confirm be asked to draw diagrams and then measure the magnification. Okay, and the thing you have to note there is put it to 1 dp and the unit is X. Planning, okay, the planning of experiments, you can always pick some information from the actual experiment that you did and then use it for your planning. Okay. And also remember the graph and why finding the x-intercept for this question is important. Okay, and then reliability. To make the experiment more reliable or more accurate, you must always repeat it. Okay, and then calculate the average. And then again, the concepts of osmosis, you all should know it by heart by now. Okay, it's something that will come out, so remember to study for that. Okay. Okay, so that's the end of the going through session, the mini session. Okay, and I hope that y'all learn something, y'all take away something from this short video clip. And yeah, just good luck for your O levels. Keep calm and just read the questions properly. Okay, thank you. Bye bye.